Okay, so today we're going to talk about our three classes of hormones. And so we're going to start with our first one, which is the amines. And so here, these are our amine hormones. We have our T3 and our T4, which are thyroid hormones. And then we have our norepinephrine, epinephrine, and dopamine, which are our catecholamines. So our T3 and T4 are nonpolar and our catecholamines are polar. So we have to be conscious of that there's a few different groups within amines itself. So this is kind of unique, but it makes sense because if you look at the structure of our epinephrine and norepinephrine and our dopamine, they have an extra OH group, which sets them apart and makes them a little bit more polar. And our thyroid hormones have a bit more of that ring nonpolar structure. So if we move down here to our peptides, we see that they are kind of similar, but they're going to be polar typically. And these are some examples right here. And then down here, they are released by exocytosis, and it's regulated by that. So it's regulated by our exocytosis because they are constantly made. So once they are regulated by the exocytosis, that's how it controls how it gets out into the system. Because once it's out there, it can be used. So this is how they like regulate that release by these secretory vessels that leave from the Golgi. So, because they are polar, they can't get through the cell membrane, so they bind to surface receptors on the outside of the cell. So, they bind to the outside of the cell, whereas our next category, when we go in, our nonpolar ones are going to bind inside. And they can be degraded quickly here as well. So, if we go to our steroids, these are a little different. So, they are all nonpolar. And so here are some examples up there. And because they're nonpolar, then we know that it will, because it's the opposite of our peptide ones from before. So because they're nonpolar, their receptors are in the cell because it can get through that membrane. And yeah, so these are all definition or derivations of cholesterol. So we can see that they all are pretty similar structure-wise to our base here. So moving down here, we have most amine and peptide hormones are polar or hydrophilic and are carried in the blood to their targets, except for our T3 and T4, because those ones are going to be our nonpolar, our thyroid hormones, which are right here. So then we have down here our steroid hormones and thyroid hormones are bound to carrier molecules in the blood. So they are bound to like globulins and carrier proteins so that they don't just immediately dissolve into each cell because so their cell receptors on inside of the cell, so they are regulated by carrier proteins. So a little different, but if they weren't regulated by those carrier proteins, then they could just dissolve into any cell and just act at any time. So this is how we kind of regulate that. And then if we move down here, we see how our cell surface receptors, like our peptides and our amines, our polar ones, are going to bind to like the cell membrane and then work on the inside of the cell through second messengers or changes in membrane potential. And then down here, we see that our hydrophobic ones are going to bind inside the cell. So this is a good diagram showing how it moves throughout the cell, and it can either bind into like the nucleus or the cytosol, but it can get through. So if it goes into the nucleus um, and like into the cytosol, it's going to have nuclear effects. So it's going to right down here, I wrote like regulate transcription activities. So like it's going to work on the DNA. So DNA changes through like transcription and such. So yeah, those are our three different types of cells and kind of how they work, a basic overview of them.